Hello everyone. We are going to start a new chapter from the physical pharmaceutics 2. The chapter, it is the drug stability. So the thing is that why we need to study the drug stability. We all know that the drug, they are supplied to the patient in the form of different formulation, isn't it? Sometimes we used to give the drug as a tablet. Sometimes we used to give the drug as a capsule, sometimes syrup, sometimes as a suspension, emulsion, whatever. So after the preparation of this formulation then takes some time to reach out the patient, right? And from the time the drug has or the formulation has reached the patient till the complete consumption of that formulation, the formulation, it needs to be stable. That is the stability of the drug. So in this chapter we are going to deal with the different attributes which affects the drug stability. Moreover, how we can control the stability of the drug. So first of all, let's have some basic example of stability, some basic definition of stability. So stability, as per the United States Pharmacopoeia, that is the USP, it defines the stability of the pharmaceutical product as the extent to which a product retains within specified limits and throughout its period of storage and uses. So that is known as the shelf life. Means the shelf life can be defined as a period till which the drug or the formulation, it will retain its property. Means it will not going to, or it is not going to get degraded. It is going to show its pharmacological action, right? So the capacity, or you can say the capability of a particular formulation, whatever type of formulation, tablet or capsule or any type of formulation, right, in a specified container. Suppose we have stored the tablets in the blister package or in a simple container, suppose, right? So formulation in a specific container to remain within a particular chemical, microbiological, therapeutically and toxicological specification. Means I have put the paracetamol in a particular container. The pharmacological action of the paracetamol is to reduce the temperature. That is, it is acting as an antibiotic. Means till the time the drug or the formulation will act as an antibiotic, it is regarded as stable, right? So the drug stability is defined as the ability of the pharmaceutical dosage form to maintain the physical, again chemical, therapeutic and microbiological property during the time of storage. So these are actually different definition, right? So different people used to give different definition of the drug stability based on their perspective, right? So the last definition is nothing but the purpose of the stability study is to provide the evidence on how the quality of the active substance, that is the drug, it get affected. That's why we are going to perform the stability study, just to see what are the factors which affect the stability of a drug. Done. So importance of stability. So you see, the chemical physical degradation of the drug substance, if it happens, if the drug substance is degraded chemically as well as physically, it will affect what? It will hamper what? It will hamper the pharmacological effect. So which is then affecting, if the pharmacological effect is affected, if the pharmacological effect is distorted, then it will have effect on its therapeutic action of the drug as well as its toxicological effect also, right? So the pharmaceutical products, they are used therapeutically based on their efficacy and safety. So whatever the pharmaceutical product we use in our day-to-day life just to alleviate the disease from our body, they are generally based on their efficacy and safety. If the drug is not stable, if the formulation is not stable, then definitely it will affect the efficacy and safety, and thus the degradation of the drug will take place. So maintenance of the quality until the time of uses or until their expiration date. So the drug stability is important just to know the quality of the drug till the time of usage, means from the time you have prepared the drug in the or the formulation in the industry till it's reached the hand of the patient. Done. So the quality should be maintained under various conditions, because we know that after the preparation of the drug in the pharmaceutical industry or the formulation in the pharmaceutical industry, it is subjected to be transported. Suppose we have a pharmaceutical unit operation at, suppose, New Delhi. From New Delhi, the drug will come via air or via road or via railways, right? It will reach our state. From our state, it will be sent to the respective district. From the district, it will be sent to the respective town. From the town, some retail shop will sell the drug and it will go in the hand of the patient. During this time, our motto, our intention, or the intention of this stability study is to just to confirm the stability of the drug. So adverse effect of instability of the drug. Suppose if the drug is unstable or it becomes unstable, so what may be the consequences? Number one, loss of active drug. Sometimes the active drug, the active pharmaceutical ingredient, that is the API, it may get degraded. Example, aspirin, it get hydrolysis, and oxidation of the adrenaline. These are the example. So next example, it is the loss of vehicle. Vehicle means we all know that vehicles are the additives which are used in the formulation. So loss of vehicle. For example, if we have kept the emulsion, suppose the emulsion of oil in water type of emulsion in a higher temperature, what will happen? The water may get evaporated, right? Evaporation of alcohol from the alcoholic mixtures. Yes, because alcohol, it is volatile in nature. Next, loss of content uniformity. Content uniformity means what? Throughout the suspension, throughout the cream, throughout the semi-solid preparation, wherever it is applicable, the content of the drug is same everywhere. But if the drug is unstable or if some physical parameter has acted on that, this can lead to the loss of content uniformity. You will not get uniform content throughout the tube of a, suppose, cream, or throughout the bottle of an emulsion. Example, creaming of emulsion, impaction of suspension. So these are example of loss of content uniformity. Next is the loss of elegance, appearance. That is, sometimes fading of tablets. The color of the tablets may fade, or sometimes colored solution may get faded. Next is the reduction in the bioavailability. This is our concern. So if it happens, then it means if the drug is not stable, then it can lead to the reduction of the bioavailability. The drug may not be available in the body after taking the drug. Aging of the tablets resulting in the change in the dissolution profile actually. So the next one, it is the production of potential toxic material. Yes, it happens sometimes. So the drug or the API may get converted into some sort of toxic chemical, which may also affect the patient health. Then, so what is the need? You see, need of stability study, it is required because number one, there are various factors. You see, quality varies with the time. We know that initially we may get a very good quality, but if we have stored it for a long time, after the time the quality may be affected. Thus, it is necessary to study the stability. Next, it is safety point of view of the patient. We have to check. Suppose we have prepared a drug in the Delhi, it will reach to some person. Some person in some area of, suppose, our state, he is going to take this drug. We have to determine at the time when the patient is going to take the drug whether it is stable. Next is essential quality attributes like your temperature, light, etc. Prevention of expenses. Because due to formation of toxic product, the patient need to be hospitalized, or sometimes all the batch of the drug may degrade. We have to reformulate everything. Next, it is the container closure stability. After closing the container which contains, suppose, the formulation, we have to check the stability. Next, storage condition in which the drug will be stored during the time of transportation or during the time at the retail shop, and the shelf life of the drug product. So these are the defined attributes which actually emphasize the need of the study of the drug stability. So where the stability study takes place? Where the stability study actually takes place in the drug discovery and development phase. So you see there's a simple chart. First of all, what we used to do in the drug discovery process, first of all, a new chemical entity it is find out. We will find a new chemical entity. Suppose a new molecule we have discovered, right? First of all, preclinical studies are done. The preclinical studies of the drug includes, we have to check the chemistry of the drug, we have to check the physical properties, then the biological properties. In the biological property, we have to check the pharmacology, ADME. ADME, it is absorption, distribution, metabolism and excretion, or you can say elimination. So this is a part of pharmacokinetics. Then we are going to check the toxicology. Then we are going to make the formulation, tablet, capsule, whatever it is. So before making, we have to check the pre-formulation. In the pre-formulation, what we are going to check? Bulk characterization of the powder drug will be done. Stability analysis, it is done. Then solubility analysis. So here you see stability analysis, it is highlighted because it is an important parameter in the pre-formulation stage of the drug development. Afterwards, we used to perform the clinical studies. That is the drug that we have, the molecule that we have developed, it will be tested on the subject, that is the patient. And finally, we used to perform the post-marketing. Means after it has reached the patient has consumed the drug, then we are going to check the effect of the drug later on. Means after consumption, if the drug is showing any sort of action, right? So the next thing is the type of stability. You see, type of stability. So which are the factors which affect the types of stability? That is the number one: chemical, physical, therapeutic, toxicological, microbiological and genotoxic. So these are the different attributes. Right? So types of the stability testing, basically we have three types. Number one, it is the long-term stability testing. It is also known as ambient, whereas the storage condition is, these are the storage condition. We are going to store the drug at a 25 degree plus minus 2 degree with a 60% relative humidity plus minus 5%. Testing time, sometime at the 0 month we are going to check, then after 3 months we are going to check the property, then 6 months, then 9 months, then 12 months. In such a way we are going to study the formulation till 60 months. So but the minimum time period it is covered, it is the 12 months actually. Till now, we generally used to perform till 12 months. It is based on the molecule. If the molecule is new, we are going to perform it till 60 months. The next one is the intermediate one. So it is also known as the control one. So here the temperature is 30 degree plus minus 2. Relative humidity 60% plus minus 5%. It is performed generally, it is subjected to perform generally till 12 months, but for the convenience we used to perform till 6 months. The last one, it is the accelerated stability study. Here the temperature it is raised, you see, 40 degree plus minus 2 degree, and generally it is performed till 9 months. So for convenience we used to perform it till 6 months. Then so degradation part 4. So the degradation of the active drug actually, that is the API, active pharmaceutical ingredients, actually it sometimes led to the lowering of the quantity of the therapeutic agent in the dosage form, right? If suppose we have added 500 mg of paracetamol, but suppose the tablet it gets degraded, that tablet may not have 500 mg of paracetamol. Suppose from there 150 mg of paracetamol has degraded. This type of effect actually happens. The next one, it is the toxic product formation. As I have already told you that due to some sort of reaction the toxic product may forms, right, due to the decomposition, and this can affect the bioavailability of the drug within the patient body. The next one, it is the change in the physical appearance of a given dosage form may take place. As I already told you, the fading color of the tablets, fading color of the colored solution, these are actually change in the physical appearance. Suppose creaming, it happens in the emulsion, right? The next one, that is the degradation may increase or decrease the potency of the drug, right? So type of degradation pathway, again these are the pathway, degradation pathway: physical, chemical, microbiological, toxicological and therapeutic. So these are a degradation pathway, right? So let's discuss some of them. Physical degradation, example: loss of volatile compound. If heat is available, then the volatile compound may come out, may escape. Next one, loss of water. Even the temperature, if it is high, then water may be lost from the formulation. Then absorption of water. Sometimes water may get absorbed in the product, which will also affect the quality of the drug. If the water is absorbed, definitely the chances of the growth of the microbes increases. If the growth of the microbes increases, then definitely the chances of decomposition of the active pharmaceutical ingredients, it increases, right? The next one, it is the crystal growth. Crystal growth, it appears when, if the solution it is leaving or losing the water content, then growth of crystal of the drug, it takes place. The next one, it is the polymorphism. Polymorphism, I hope you people know. Polymorphism means a simple drug, it will have 2 to 3 different types of structure. That is a polymorphism. So one structure may get converted into another structure. Now the reason is that you might ask me one question, that why, as the compound is same, what is the effect of polymorphism? Yes, the polymorphism have the effect, as we people have already studied the chemistry. In the chemistry, we know that if the structure of the molecule it is changed somehow, the property of the molecule get changed. That is, we study all these things in the medicinal chemistry. That is SAR, structural activity relationship, right? Next is the color change, which is actually easily observed by the naked eyes. Then the next one, it is a photolysis. Photolysis is nothing but it is the degradation under the light. So let's study the loss of volatile components or compounds. So some volatile components like example here, alcohol, ether, iodine, volatile oil, menthol, camphor, whatever it is, they always have the tendency to escape from the formulation and lead to the degradation. Example, some types of the tablets, like example your nitroglycerin tablet. So nitroglycerin tablet, have you heard? So nitroglycerin tablets are generally used in the treatment of angina pectoris, is the chest pain. So these tablets are generally sublingual tablet. They are put in the sublingual area. The next example, it is your aromatic water. In aromatic water, we know that water is there and we have some sort of chemicals which have aroma. The volatile component, the volatile oil may escape from the aromatic water. Prevention, how you are going to prevent it? Simple. Such products should be placed in a well-closed container and temperature should be proper. Means as per the instruction given by the manufacturer. The next one, loss of water. So loss of water, generally it happens in oil in water type of emulsion. If it happens, then what will the consequences? Stability. Sometimes crystallization of the drug also takes place, which actually affect the potency and it can lead to the decrease in the weight of the formulation. Example, the water, it evaporates from the borax, sodium sulfate, and in the cream, especially you see oil in water type, they become dry by loss of the water. If we store a tube of cream for a long while, you can see that after putting pressure over the tube of the cream, you will find that cream, it is quite dry, right? Quite dry. It is because due to the loss of water. Prevention, products should be placed in a well closed container, right? The next one, it is the absorption of water. Hygroscopic drug. So hygroscopic drug, they generally absorb water from the environment, and if it absorbs the water, definitely, as I have already told you, it can lead to the physical degradation. Effervescent powders and tablets, they will deteriorate if we store them in a moist temperature. Effervescent powders, example, Pepfiz, and effervescent tablet, example, Dispirin. So Dispirin is there, some antacid preparation are there. If they are stored in a moist area, their property of effervescence, it is lost. Example, your powder, example, liquefaction and degradation may occur as a result of absorption of the water. So apart from that, suppositories, right? Suppositories, they're generally made up from hydrophilic substances like glycerin, gelatin, polyethylene glycol. So their consistency to form or become jelly-like appearance it lost if the water is move out, or it is lost if the water is coming in. Means they should be maintained as per the instruction given by the manufacturer. Prevention, simply the such products should be stored in the well closed container in a dry place. The next one, it is the crystal growth. How crystal grow? So drug, when it will lose the water, it has a tendency to become crystal. Thus the crystal growth occurs. So crystallization actually it enhances in the porous tablet, and in solution it can form after in solution after supersaturation crystal growth it occurs. So example is injection of the calcium gluconate. They have the tendency to form crystals. Apart from that, in suspension, crystals generally they settle down and the caking occurs, and the suspension become unstable. Even you can see the example of crystallization even in the ophthalmic preparation. Those are meant to be used in the eyes. Prevention, simple. The solution stabilizer are added. In suspension, incorporation of the surface active agent, that is the surfactant, it is to be done, and by increasing the viscosity of the suspending material, crystal growth can be prevented. Done. So thank you everyone for your patience hearing. If you liked the video, just press the like icon. You can share this video and you can subscribe to my channel. Thank you once again.